sequence of events that needs to occur for muscles to physically contract. Okay? And so the first step that we began to talk about was that the front lobe of our brain, the frontal lobe of our brain, which is covered by the frontal bone, is your motor cortex, and its role is to control skeletal muscle activity in your body. And the way your nervous system communicates with the rest of your body is by creating changes in membrane potential that we talked about in lab yesterday. So creating a depolarizing event followed by a repolarizing event. And because muscle cells, skeletal muscle cells, lack gap junctions where cells can communicate with each other, then skeletal muscle cells are independently isolated, and each cell has to have a connection to the nervous system. So the muscle, the nerves that go out from our central nervous system out into our body that actually control skeletal muscle activity are called somatic motor nerves. And where they interact with a muscle cell is actually called a neuromuscular junction. Okay. So what we want to do is we want to look at how a nerve interacts with the muscle cell at the neuromuscular junction. So at the top, this would be our nerve fiber, which would be part of a nerve arising from your spinal cord and going out to your body. And then at the end of the nerve, what we have are what we call axon terminals. And so there's kind of a branching pattern with these axon terminals that actually create the neuromuscular junction, which is the connection between the nervous system and the muscle cell itself. So what really happens is when you look at it with a fine microscopy, we realize that the nerve is actually touching the muscle cell itself. And we actually have a modified membrane where the nerve is overlying the muscle cell that we call the motor endplane. Okay? So we end up with a space that we have to communicate across. And so a word we commonly use for a space in anatomy is a cleft. And so the interaction between the nerve and muscle is a synapsis. So the space is a synaptic cleft. And so because we have this synaptic cleft, then we actually need a compound that can be released and diffused across the space. So the common name we use for compounds that allow for one neuron to interact with another neuron, or one neuron to interact with something like a muscle cell, are neurotransmitters. And if you'll recall, when we went through skeletal muscle, the neurotransmitter that was used for skeletal muscle was acetylcholine or acetylcholine. All right? And so what happens is we've got this wave of depolarization that was created by your brain that goes out via a nerve. And when it reaches a synaptic impulse, it triggers the release of acetylcholine from the structures that are called synaptic vesicles. So what actually happens is the neuron's nucleus has the gene to code for the formation of acetylcholine. Acetylcholine is produced on ER and then packaged by Golgi apparatus. And then it builds up in the synaptic impulse in these vesicles that were created by the Golgi apparatus. And if the neuron is not stimulated, then the vesicles are just retained in the cytoplasm of the synaptic impulse. So what we have to do is we have to trigger their release by creating a depolarizing event that was initiated with your brain. And so these synaptic vesicles will move to the surface of the synaptic impulse, and the acetylcholine will be released, and then it'll diffuse across the synaptic cleft. So diffusion always requires a compound to be more concentrated in one area, less concentrated in the other area, so that the compound will diffuse from an area of higher concentration to an area of lower concentration. So as these synaptic vesicles release acetylcholine, and it diffuses across the synaptic clay. Now, the part of the muscle cell, which is called a motor end plate, contains membrane-bound receptors that the acetylcholine has to bind with. So we have to have a unique receptor that the acetylcholine is going to bind with. And it just so happens that these receptors are modified sodium channels. And as acetylcholine binds with the receptor, that allows sodium to enter the muscle cell itself. And then for very discrete, rapid communication, a neurotransmitter cannot exist for a long period of time. Otherwise, you lose the ability to communicate. You just have a long communication, kind of like a record playing itself over and over and over again. So we actually have an enzyme that quickly breaks down acetylcholine so that it doesn't stay in the synaptic cleft very long. So that if your brain needs to communicate with the muscle again, it actually has to send another depolarizing event and cause the release of more acetylcholine. So it's very important that this acetylcholine doesn't hang around very long. It's quickly broken down. So we have very discrete, rapid ability to actually communicate. All right. So the thing to think about that we talked about yesterday in relationship to this is that we had a membrane that had 3 phases, so at rest. And at rest, our 2 channels that we're going to talk about are sodium channel and our potassium channel. Are closed. So both are closed. And we have this active transport mechanism called an antiquarter that is constantly moving sodium out of the cell and constantly putting potassium into the cell. And if you look at the periodic table, sodium and potassium are different size molecules, and so so sodium is more likely to move passively through the membrane because it's a smaller molecule. So we're removing basically 3 sodium for every 2 potassium to explain the leakage difference in it. Okay. And because all the channels are closed and we're maintaining this membrane potential so that we have a higher abundance of potassium inside the cell in the cytoplasm. And we have a higher abundance of sodium outside the cell in the interstitial fluid. Then the membrane potential stays the same as the -70. And if we were using a machine that graphs changes in membrane potential, then the machine would give us a flat line. Because no change is occurring. So what actually happens is, because acetylcholine is released and binds with these receptors and opens a sodium channel, we have a mechanism because of the release of ACH or acetylcholine that we can move the membrane from -70 millivolts to up -55. Okay. So, and what we said yesterday in lab is this is what we would call a threshold event because we're moving the membrane from -55. So the take-home message is that the critical role of acetylcholine is to allow the membrane to reach threshold. And if we can move the membrane to threshold, then what will happen on the membrane is we'll get this depolarization event. Okay. So let's look at the next step and then put the next things together as well. Right, so one thing we're going to do in lab on Wednesday, tomorrow, is we're actually going to look at muscle tissue, and it'll give a chance to review the characteristics of smooth muscle, skeletal muscle, and cardiac muscle that we looked at. And one of the things we talked about yesterday was the way in which skeletal muscle works is we have this all or nothing principle that says that at the sarcomere level, at the myofibril level, at the muscle fiber level, and at this level we call a motor unit, that either contraction occurs or does not occur. There's no such thing as a partial contraction. So the way that we actually control the amount of contraction that occurs in a muscle like our biceps is the muscle is made up of these subunits called motor units. And what we talked about in lab yesterday was that a motor unit was all of the cells controlled by an individual nerve. And if that nerve causes this threshold to occur at all the cells, then the cells will physically all contract as a unit. So we call it a motor unit. That's what you're actually looking at here, is part of a motor unit under a microscope. So these are skeletal muscle cells with the A bands and I bands that we've talked about in the past. This is actually a nerve with its branching pattern, and this is a neuromuscular junction. So we have going over to this cell, this cell, this cell. So what we're actually seeing is a neuromuscular junction that's part of a motor unit. The little tiny dark things here are actually the synaptic vesicles of acetylcholine. And so if we get a nerve potential, a depolarization on this nerve, it triggers the release of these that cross the synaptic cleft and then trigger this threshold effect on the skeletal muscle itself. Now this is a light scope, and with the light scope we cannot actually see the synaptic cleft. It would require us to have a picture of an electron micrograph that would actually show us that space, because the space is actually darn small, really small. Okay. So the next step then is that this action potential causes the opening of the sodium channel. And to do that, what we have to do is reach this threshold. And then once we open sodium channels, the sarcolemma, which is the modifying membrane around the muscle cell, is going to undergo this depolarization pattern. So if we kind of think about that, then what we would have is a muscle cell with its A and I bands. Then we have this neuromuscular junction somewhere along its length. And so this threshold effect is actually going to occur at the neuromuscular junction. And if we reach threshold, because these channels are called voltage-gated channels, changes in voltage activate them, and what we're going to do is we're going to open our sodium channels. Music on the video. So then we have sodium that's in higher concentration outside the cell. So what's the sodium going to do? Diffuse into the cell. Now the other thing we didn't talk about over here is that in addition to the potassium that we had in our cytoplasm, we also have cellular proteins that have basically a negative charge on them. So that during this period of rest, the membrane has a positive charge to the outside and a negative charge to the inside. And that's where this minus 70 actually arises. So at the neuromuscular junction, when we open the channels with acetylcholine, we move the membrane here to reach threshold at minus 55. And then that opens sodium channels. And then that event will occur in both directions from the neuromuscular junction, where we're going to start opening sodium channels. And what's going to happen is, because sodium is going to rush in, we're going to continue to move our membrane away from the minus 70. And what we're actually going to do is move it to 0, where everything's equal across the membrane, and then eventually a little beyond 0 to plus 30 millimoles. And so the membrane is actually now going to reverse its charge. So we have a negative on the outside of the membrane and a positive on the inside of the membrane. And that's what we refer to as this depolarization event. Okay? And then it's going to move down the muscle fiber or cell from the neuromuscular junction in both directions. So this is essentially a graph of what we, part of what we were just doing. Okay? And this is a graph that's a more detailed graph that's related to the blue graph we talked about yesterday that was under the myograms for a simple twitch and wave summation and unfused tetany that we talked about in lab. So if we review what we said, is if the membrane is staying at minus 70 and the membrane is not changing potential, then it would graph as a flat line, because the instrument we would use would actually would respond to changes in membrane potential. So if we release acetylcholine and acetylcholine binds with the receptors in the motor end plate, then we're actually going to move our membrane from minus 70 to minus 55. So it moves upward. And that is what we call a threshold event. So yesterday in lab, I was using electricity to create the threshold instead of acetylcholine. So I was taking over the role of the brain to make muscle contract by using electricity. Okay? So once the membrane hits minus 70, then we're going to open sodium channels, and the membrane is going to go through a very rapid depolarization. And so the depolarization would actually be graphed by the machine as an upward swing by a physiograph as an upward swing. So we get this upward swing. And we're going to look at that in lecture 3 again. And we're going to look at the graph of a myogram relative to the graph of membrane potential and look at how those look together. Okay. So what we're going to do is get this rapid depolarization event that's actually going to occur. So this graph is actually for a neuron, a motor neuron. So this is actually a motor neuron, and this part of the neuron would be in your spinal cord, and then this part of the neuron would exit the spinal cord and become part of a spinal nerve going out to your body, and then this would be a motor unit. And every one of these endings would be associated with a single muscle cell. And so all of the muscle cells that this particular motor neuron would interact with are defined as a motor unit. And a motor unit will either contract fully if it's been stimulated or not respond at all. But you get no such thing as a partial response or contraction. Okay? And so what we're looking at with just the tube, with the microscope, was like the two cells that would be attached to these two neuromuscular junctions but we would have cells with every neuromuscular junction that we're seeing. And in muscle cells behave the exact same way. Muscle membrane cells behave the exact same way as this neuron membrane behaves, in the sense that we have a threshold, we have this resting membrane potential, and we go through this very rapid depolarizing phase. So what I want to do is I want to complete our picture for us. So these channels respond to changes in membrane potential. So they're voltage-gated channels, because that's what causes them to occur. So what happens is the membrane moves to plus 30, is as soon as the membrane hits plus 30, then what that's going to do is close our sodium channels. So this plus 30 causes the sodium channels to close. So what actually activated it was a movement toward minus 55. And then what eventually closes them is when the membrane hits plus 30. And as soon as the membrane hits plus 30, the sodium channels close and potassium channels open. So we're going to close sodium channels, open potassium channels. And that sets us up for our next phase of our membrane, which is a repolarization event. Okay? So what's going to happen, because potassium was in higher concentration inside the cell, and potassium is going to diffuse out of the cell itself. And what's going to happen is now we're going to move the membrane back toward minus 70. So the membrane is going to move back toward minus 70 millivolts. And as it does that, then it's going to become positive on the outside again and negative on the inside. So anytime we're dealing with a membrane whose channels, ion channels, are voltage gated, then they're going to open and close quickly based upon relative change in membrane potential. So if the membrane never changes, the membrane would stay at rest. All the channels would be closed. And what maintains the abundance of ions inside and out, sodium outside, potassium inside, is this antiquarter called the sodium-potassium pump. And this is always active, as long as you have ATP to drive it. So this would fail if you couldn't make ATP.
Yesterday you said negative 90. Is it interchangeable?
No, I was actually change it to 70. It's negative 90 on cardiac membranes, but not skeletal membranes. So I was just confusing my membranes. But it's the same idea across the board. Okay. So if enough acetylcholine is released to move the membrane to minus 55, we get a threshold event, and the membrane goes through this rapid depolarization. So as soon as the membrane hits plus 30, sodium channels close, potassium channels open, and the membrane undergoes repolarization. So membrane changes are very, very rapid, going from rest through a depolarization and a repolarization. And a repolarization takes us full circle back to rest. So I want to make that point. So notice that, again, I would give the author a bad grade because they didn't give you any units as to what our milliseconds are. But notice that this graph is milliseconds going this way, and membrane potential in millivolts going that way. And so notice that from the point of stimulus to the time that we are back at a resting potential is only 2 units within this millisecond time frame. Okay. So very, very, very rapid. Now there's a couple of ideas inherent in what's going on here that are going to become more important when we actually look at neurons in the next unit, and not quite as important with skeletal muscle physiology. And so we're going to come back in the next unit, talk about what this absolute refractory period, relative refractory period are all about in relationship to membrane potential, where it's more critical to the way the membranes behave. All right, so just to reiterate what we put on the whiteboard, we would start at a resting potential where all gates are closed, so no sodium, potassium removed. That would graph as a flat line on any graph. And we're going to eventually put this together because in the next unit we're going to talk about electrocephalograms that we do clinically to determine somebody's brain function. And we determine somebody's brain dead if the electrocephalogram is creating a flat line because there is no change in membrane potential going on in the neurons of the brain. And then when we get to EKGs, where we're going to look at this same event in cardiac muscle, then on a flat line in an EKG, it also means there's no change in membrane potential. So if you'd had a heart attack and they put an EKG on you to see what's going on, and all they're getting is a flat line, you would be pronounced dead because you have no change in membrane potential going on in your heart. Okay? So it graphs as a flat line. So if we reach threshold, and what triggers threshold in your body would be the adequate release of acetylcholine at the neuromuscular junction. And if we move the membrane from -70 to -55, then we open sodium channels and we get this rapid depolarization event, which is graphed as an upward swing by the physiograph. As soon as we reach +30, because these are voltage-gated channels, the sodium channels close, potassium channels open, and the membrane quickly undergoes a repolarization. And so in a repolarization, what we're going to do is move the membrane from +30 back to -7. And so through the repolarization, our membrane is going to go back to -70. And then once we reach -70, then we convert to a resting membrane potential again. So it's a real cyclic pattern that occurs on membranes. All right. So the next step is thinking about how a muscle cell is constructed and how we can take this surface event on the sarcolemma of the membrane, and we can communicate it deep into the cytoplasm of the membrane. So we have to communicate this depolarization event from the end of the cytoplasm, and that's what T-tubules are about. So remember, T-tubules were downward folds of the membrane that penetrate down into the cell itself. And then what we're going to do is we're going to transfer the depolarization of the T-tubules into the depolarization of the sarcoplasmic reticulum. So remember, the sarcoplasmic reticulum was unique in muscles in that it stores calcium, and that it's designed in a way so that following T-tubules down into the muscle itself are these parts of the sarcoplasmic reticulum we call terminal cistern. And that's where the calcium is actually stored. So what we're going to do is we're going to use the depolarization to open a third channel that we haven't talked about because they're deep inside the cell, which is a calcium channel. And that's going to cause calcium to be released from storage. So again, if we think about it, what we've been talking about is the fact that we get this depolarization event traveling on our sarcolemma. Skeletal muscle cells are really quite large cells in terms of their internal volume. So we have these downward folds in the membrane called T-tubules that penetrate down into the cell. The sarcoplasmic reticulum lines up in relationship to the T-tubules, so that we got these blocks within the sarcoplasmic reticulum that we call terminal cisterns. And the terminal cisterns are where we store calcium. So as we move the depolarization event from the surface down into the cell via T-tubules, the depolarization is transferred to the sarcoplasmic reticulum, and we're going to open up calcium channels. And calcium, because it's more concentrated in the terminal cisterns, is going to diffuse out and interact with these thin filaments and protein associated with the thin filament. Yes?
Calcium channels voltage-gated?
Yes. They are voltage-gated as well. That's why the depolarization triggered it. Right? So the released calcium becomes important because it's going to bind with this protein molecule called troponin. And then troponin and tropomyosin interact to create a complex. And what they typically do is they cover a critical site, which is called the cross-bridge binding site or the myosin binding site on our actin molecules. So that requires us to do another real quick review of the structural components that we're seeing within a sarcomere. So remember, a sarcomere went from Z line to Z line, and the Z line organized these actin molecules. At the center of the sarcomere, where an A band is, we have myosin molecules, and they're organized by an M line at the center. When you look at an A band, it stays darker out here and lighter in here, so this was called the A zone. And it's darkest where we have overlapping actin and myosin molecules. And to make a muscle contract, what we have to do is change the shape of the troponin and tropomyosin molecules, because they're covering these dark brown dots on our actin, which are sites where actin and myosin can interact. So as we depolarize the membrane, and the membrane carries the depolarization down to T-tubules, then it's transferred to our terminal cisterns. That opens our calcium channels and allows calcium to diffuse out. As calcium diffuses out, it binds with the troponin molecules. Because you're binding a +2 ion, you're going to change the way in which electrons are behaving in the molecule. And remember that covalent bonds are created by the sharing of electrons. And if you can change the way in which electrons are behaving in covalent bonds, you can actually change the shape of the molecule. So let me give you an example of that in your body, typically that you could see more readily. So when your hemoglobin gives up oxygen at your tissues and absorbs carbon dioxide, it actually changes its shape and becomes blue. So that when you look at veins, they appear blue. As soon as that hemoglobin is exposed to oxygen, oxygen replaces the carbon dioxide molecules. The hemoglobin molecule changes its shape, and blood looks red. And that's all just changing the shape of the hemoglobin molecule and changing the way it's interacting with light. That changes it from blue to red. So what we're doing here is we're taking troponin and tropomyosin, we're changing its shape so that it actually moves away from these myosin binding sites so that actin and myosin can interact. And as soon as actin and myosin can physically interact, the muscle will begin to contract. Okay? So all the steps we just talked about, from the point where the membrane became depolarized to this point where calcium has bound with troponin and tropomyosin so that actin and myosin can interact, all occur during that time frame that we talked about yesterday in lab, which is a latent period. Because unless actin and myosin can physically interact, you can't contract. Okay? All right, so then the next three steps are all part of a contractile pattern that repeatedly occurs during a contraction. So that myosin cross bridges bind to the cross bridge binding sites on the actin. We have an enzyme, and there's a pattern that we use in biochemistry where all new enzymes end in ASE. So I just, instead of writing out adenosine triphosphatase, I just put ATP ACE as an indication that it's an enzyme. So we actually have an enzyme that allows us to take ATP, convert it to ADP plus phosphate. And anytime we do that, we release energy. So if you go back to Bio 160, one of the things that you talked about were the laws of thermodynamics. And within the laws of thermodynamics, there's the statement that anytime you transfer energy from one form to another form, there's never 100% conversion, and you always give off heat energy. It's the heat energy of skeletal muscle that maintains our body temperature. When we're converting this ATP to ADP plus phosphate. And the amazing symmetry in the world around us is from bacteria to humans. We all use ATP as an energy source, and we all have this enzyme, which means we share a gene on DNA to manufacture the enzyme throughout the biological world. Okay? All right. So the kinetic energy, so remember potential energy is energy, stored energy. So ATP contains potential energy. As soon as we take the phosphate off and convert it back to ADP, we take the potential energy and convert it to kinetic energy. So the kinetic energy causes a power stroke to occur, which is actually going to pull the actin toward the center of the sarcomere. Okay. And then this is going to occur in every sarcomere, in every myofibril, within the muscle cell itself. And then it's going to occur in every sarcomere and every myofibril within all the muscle cells that are part of that motor unit. So they're all going to work together. Okay. All right, so the key to it is that we have to have actin and myosin interacting. And we actually have to have an ATP that can bind at an ATP binding site. And what we're going to do is we're going to use the enzyme to convert ATP to ADP. As that happens, we release kinetic energy. And kinetic energy is essentially going to be used to cause the myosin head to rotate toward the center of the sarcomere. And as it does that, it's going to pull the actin toward the center of the sarcomere, which results in the contraction that we can physically see. So just as a quick review, remember a sarcomere runs from one Z line to the next Z line. So it's the middle of one I band through a band to the middle of an I band. So when a muscle is relaxed, we have a fairly thick I band and a fairly thick A band. As a muscle contracts, the I band is going to begin to disappear because the Z line is going to be pulled toward the center of the sarcomere. Okay? So if you take that picture and then you think about it. Now, there's something that it's just a choice of words that I think confuses students. So what I'd like you to do is take the word partially contracted muscle out. Because I just told you what? Muscles cannot partially contract. So it's just a matter of semantics. But initially contracted muscle. So what we're looking at is a relaxed muscle, a muscle that's just beginning to contract, and then the muscle as it goes from initial contraction to full contraction. So within milliseconds, we're going to make this change. So remember that when the muscle is completely relaxed, we have the A band, we have the I band, we have the A band. Again, the I band at the center has a Z line, and it's very light because we only have actin molecules. As we enter the A band, we have this overlapping zone of actin and myosin. Then we have a zone where the actin is by, the myosin, is by itself. So that's called the H zone. Now what's going to happen is that these cross bridges are going to connect to the actin. And as they connect, and ATP is used, they're going to pull the actin toward the center of the sarcomere. So notice that while it's just starting to contract, or initial contraction, the Z line now has gone from being over here to here. And as we go to a fully contracted sarcomere, which would mean all the myofibrils are fully contracted, which means all the muscle cell is fully contracted, which means that all the muscle cells innervated by that neuron would have done the same thing, and that's the idea of the motor unit. Then look at what happened. Now the Z line is sitting right next to the end of the A band. And so all of this that was staining light, because it only contained actin molecules, now is gone. And so what we've actually done is pull all of the actin molecules toward the center, so that now everything's overlapping, and you would no longer see a I band. Yeah. And then every sarcomere does this all in a row. Every myofibril does this all at once, so that a muscle cell does this all at once. And then every muscle cell controlled by an individual neuron does it. So that is amazing when you think about it. Yes?
I got a question about the relationship between actin and myosin. Is the myosin sort of just doing like one pull-up, or is it like crawling?
No, it repeats.
Repeats?
Yeah, it repeats. So as long as the depolarization event is occurring, and as long as calcium is available, what actually happens is, as soon as the ATP has been hydrolyzed ADP and the power stroke occurs, then the myosin releases. A new ATP binds with it. And as soon as the ATP binds with it, it binds with actin. And it repeats again and again and again and again. Yeah. So it's more like crawling instead of a one-time event. Okay, so the thing about creating this event list here is I did it in a way in which it describes the relationship of activity that has to occur within the muscle to go from initially contracting to stopping the contraction. But what you have to remember is that what we talked about in lab yesterday, what we just looked at in terms of these membrane events, is that the membrane event is very rapid relative to the physical going on with the muscle cell itself. So even though this 9 suggests that the sarcolemma repolarizes, and that step has to occur before a muscle can physically begin to relax, the repolarization has already occurred on the membrane while we've been talking about the internal activity of the muscle cell itself. Now is the key to understanding the overlapping graphs relative to that. So let's review that real quick so that we can make sense of it before we go on. So this was the graph I was referring to yesterday in lab. This is the myogram showing the latent period, contractile period, relaxation. So muscle at rest, latent period, contraction, relaxation, muscle back at rest. This is the membrane potential. So a flat line is rest. The upward swing is depolarization. And the downward swing is repolarization. So the membrane is already repolarized while the muscle is still contracting, because the events are slower within the muscle where the actin and myosin have to bind with each each other. You hydrolyze an ATP, repeat that, okay. And because to make it so that actin and myosin cannot interact, we have to take all the calcium back. Then it takes a little longer to take the calcium back. So the membrane is already gone through repolarization, and in fact it's at rest for most of the period of contraction, and most of the period when the muscle is relaxing. So membrane activity is way accelerated in time relative to the physical activity of muscles. Does that make sense? That's the important thing to understand. Yes?
Do you sign up to choose so later the same with the non-membrane rest polarization on the top correct one?
So this is myogram. So this would be latent contraction, relaxation, rest, latent contraction, partial relaxation, latent contraction, relaxation, rest. This is resting membrane potential, depolarization, repolarization, resting membrane potential. So different words, depending on whether you're talking about the physical activities, latent period contraction, relaxation, versus membrane potential, which is rest, depolarization, repolarization. Okay. This is something that confuses students a lot of times because the membrane potential, the membrane activity is so rapid relative to what the muscle physically is doing that sometimes students just think that that the membrane stays depolarized for the whole time it's contracting and it repolarizes for the time it's relaxing. That's wrong. It's very, very rapid. As soon as the membrane hits plus 30, that's the importance of those voltage-gated channels. Because as soon as the membrane hits plus 30, boom, sodium channels close. That stops the depolarization. Potassium channels open. That initiates the repolarization. Okay. So even though this sequence appears that it's like that, it was impossible to overlap the two events time-wise. All right. So I apologize if that leads to a little confusion. So all we have to do is repolarize our membrane. So as we indicated, as soon as the membrane hits plus 30, it's almost plus 30 here, it hits plus 30 here. We close our sodium channels, open our potassium channels, and the membrane enters a repolarizing phase, which quickly takes the membrane back to plus 30, which closes all channels, and the membrane goes back to rest. Now, if the muscle needs to continue to contract, the brain will send another signal. And it will initiate another depolarization event, which will create another contractile. So that was the idea of understanding the difference between wave summation, unfused tetanus, and fused tetanus. If your muscle, if your goal of your brain is to pick up and hold an object and keep bringing it upward, then the brain is constantly sending signals to the muscle and converting the muscle into a complete tetany event so that it stays fully contracted for a period of time. Okay. So again, what we're going to do is we're going to go from plus 30 back to resting. Now, what usually happens on membranes, particularly in skeletal muscle in particular, and neurons in particular, is that we actually go beyond our resting membrane potential for a short period of time. So we end up with this hyperpolarizing phase where we're now actually minus 80. Now, the only value to that for right now is understanding threshold. The threshold is unique in that we have to move the membrane to minus 55 to open our sodium channels. But if the membrane's at minus 80, now the threshold is a different number, right? Because over here, you have to move the membrane 15 millivolts plus 15 millivolts. Over here, now you have to move the membrane plus 25 millivolts. So that would require the release of a greater amount of acetylcholine. So that's the only thing about this hyperpolarization, which we say is a relative refractory proof, is that the threshold changes and the amount of stimulus that would be sent by the brain would have to be greater. So what we have to do is we have to get calcium off of tropin and tropomyosin, so that tropin and tropomyosin will return and cover the binding sites on actin before the muscle will physically begin to relax, okay? All right, so this is kind of a complete circle of what we've been talking about, all right? So what we have to do is we have to reverse what had happened. So what we've talked about is the fact that the depolarization opens these calcium channels. Calcium diffuses out, binds with the tropin and tropomyosin molecules. They move off of the actin and myosin binding sites, and then we pull the actin toward the center of the sarcomere to create a contraction. So what repolarization does is close the calcium channels. We have a calcium pump in the sarcoplasmic reticulum that quickly collects calcium and stores it back into the sarcoplasmic reticulum. So as the calcium out here drops, then calcium diffuses off our troponin molecule. And as soon as the calcium diffuses off the troponin molecule, the tropin and tropomyosin cover the actin binding sites, and that allows the muscle to relax because no longer can actin and myosin begin to contract. So the idea that we talked about was that there was this latent period. Now, latent period was where we initiated the depolarization, but we had to open the calcium channels, allow calcium to diffuse out, and then calcium had to bind to the troponin, tropomyosin. It changes its shape, now actin and myosin can interact. So the understanding that repolarization is so rapid is because it takes some time, once we've closed the calcium channels, to actually allow the calcium pump to pick up enough calcium so that calcium will diffuse off of the tropin and tropomyosin molecules, and they'll return to their original shape. So that's why when you're looking at the two overlapping graphs, the membrane has been at rest for most of the contractile period and relaxation period. Because it takes a while, once we have triggered the closure of those calcium channels, to actually sequester enough calcium back into the sarcoplasmic reticulum to get tropin and tropomyosin to return to its original shape. So that's another kind of latent period that isn't graphable like the first one.
So with the actin and myosin, once it's fully contracted, does it work its way back down as it loses calcium, or is it like pushing the release catch on a blade and having to just slide right back into the?
Well, as soon as the calcium releases from the troponin molecules, then the electrons go back to their original configuration, and the bond angle changes back.
So
it's fairly quick.
So it's a sliding more than
Right.
a. Okay.
Yes. So put this in context: when you die, the active transport mechanisms of the sodium-potassium pump and this calcium pump eventually lose the ability to do that because you use up all your ATP. And then the calcium gets released because it's no longer being held in by a pump, and it binds with tropin and tropomyosin, and actin and myosin interact, and your body goes really stiff, which is called rigor mortis. And so all of our muscles actually all contract after a time frame after you've been dead. It's all based upon the body temperature. So if you're a really cold environment, the rigor mortis event will take longer. In a warm environment, doesn't take as long. And it's because you've used up all the ATP, and you can no longer maintain these ion concentrations. And my point is, you don't have to be alive for actin and myosin to reconnect. That's kind of a little oversimplification. Okay, so what I want to do first, because we're going to begin to talk about some cool events that occur in your body in terms of the ability to move, is I want to take muscles that we've been learning and at a joint talk about how muscles kind of overlap in terms of their activities or have opposite activities. So what we're going to do is we're going to look at the elbow joint. Okay. So if we're talking about this action, which is flexion of the elbow, then the muscles that actually create the action are called prime movers. Okay. So the muscle that creates an action in a joint is a prime mover. So in this instance, then, the basic prime mover is the most anterior muscle in your brachium, which is the biceps brachii. Now, the biceps brachii is underlined by a second muscle that comes off the distal half of the humerus and actually goes over here to the ulna. So that second muscle underneath it is the brachialis. Because the brachialis has the same action as the biceps brachii, then it is said to be a synergist. Okay. So anytime you're looking at a movement, and the movement would be flexion like this, then the muscle that is initially responsible is the prime mover. The muscle that aids it is a synergist. And they both have the action of flexion. Okay. So on the back of the arm, we have this muscle, which is the triceps. It has the long head medially here, attaching to the scapula. It has the lateral head attaching to the humerus here. Then it has a medial head down here, attaching to the humerus. So it has three heads, so hence it's a triceps. It actually has the opposite action. Instead of flexion, it would do extension. So the muscle that has the opposite action in the joint is an antagonist. And to create a movement, the prime mover and the synergist have to have a stronger contraction than the antagonist. Okay. So the biceps is actually attaching to the scapula. And so the short head's attaching to the coracoid process. The long head here is crossing through the bicipital groove and attaching to the upper lip of the glenoid fossa here. So what makes it, if you have a heavy weight, what makes the muscle be able to pull the weight up? Why doesn't the scapula just get pulled forward? Because you're pulling against the scapula, and it's actually a free bone. And it's because there are all those muscles attached to the scapula that we talked about, the rhomboids, the trapezius, the serratus anterior, and they contract to hold the scapula in place. So the muscles that we contract to hold the origin in place are called stabilizers. And then their role would be to stabilize the position of the scapula. So does that make sense? So these are words that we could use at any joint when we look at what the muscles at the joint are doing. So we have a prime mover and its synergists that create the action at the joint. We have an antagonist that would have the opposite action at the joint. And then we have muscles that collectively can be called stabilizers because they stabilize the point of origin or insertion. The point of origin. They stabilize the point of origin. That allows for us to do this. So actually when you're doing a curl, you're actually using more muscles than just the prime mover and the synergies. You're actually using muscles that stabilize the scapula, keep it from being pulled forward by the the force being created. So that's step one. What do we call these muscles when we look at them in a general action? Step two is recognizing that our body works by levers and lever systems. And so within a lever system, there are three components to it. And so we'll go back to this one and then come forward. So the fulcrum is the pivot point for the movement. And so what we're going to do is we're going to use F as a symbol for our fulcrum. Right? The muscle itself contracting is creating the effort. So we're going to use E for effort as a symbol. And the resistance is what's going to be moved, and the weight of whatever we're doing is our resistance. So the way we work out is we use greater resistance than just the weight of our forearm. So if we put a 50-pound barbell in our hand, then resistance would greatly increase. So the amount of effort to create the action has to greatly increase. Yes. Okay. But the fulcrum does not change. So when we look at that, we have three types of lever systems. So a first class lever is where the fulcrum is at the center and the effort and resistance are at different ends. Okay. So there's actually a playground equipment you've probably played with, which is a classic first lever. And it's a seesaw, teeter-totter, or whatever you want to call it. Yeah. So the good example would be the fulcrum with the superior articular surface of C1, which is the axis, and the occipital convex. And then what happens is, in anatomical position, we're positioned like this. And what we can do is we can either do this or we can do this at that joint. So the action we're going to look at is this action, which is extension or going beyond anatomical position would be hyperextension. So the action we're going to look at is hyperextension of the neck. So where would the prime mover and center disc be? Are they going to be on the front of the body or the back? They're going to be the muscles in the neck. So then the muscles up here, like sternocleidomastoid, are going to be our antagonists, right? Right. So what happens then is the effort is back here, and so this is, what do you think? Is it either splenius capitis or spinalis capitis? Yeah. So attaching to the superior neutral line and then the cervical vertebrae. And the resistance is the weight of your face. And so the muscle has to create enough effort to do this. So in class 1, so a class 1 lever is E for E, F, R. So it's E for, okay. So our class 2 lever, the fulcrum now is at one end. The resistance is at the other end. Excuse me, is in the middle, and the effort is at the other end. So has anybody ever seen a class 2 lever?
Wheelbarrow.
And wheelbarrow. Yes. The wheel of the wheelbarrow, the concrete in the wheelbarrow, and your arms trying to lift the handles of the wheelbarrow off the ground. Class 2 lever. And the amazing thing about a lever system is you can lift a bunch more weight than you could without. Yeah. All right, so the example we're going to look at is going from anatomical position to this position. So that's called, what happened to my foot? Plantar flexion. or we could use also hyper extension. So what we do, unless you're, I can't ballerina thing, I can't stand on the tips of my toes. So what I do is stand on this joint right here, between my toes and the end of the metatarsals, right? So then the fulcrum is here, because that's what's actually pivoting. The weight of your body is actually sitting on the talus here, so that's the resistance. And then the muscles creating the action are these guys. And so what's this muscle? Gastrocnemius. And then this is soleus. So we would consider the gastrocnemius the prime mover, and the soleus the synergist. So what would be the antagonist muscle that we would find on the front compartment, which would have an opposite action? So the opposite of plantar flexion would be dorsiflexion. So what's the muscle that dorsiflexes are from? Anterior tibia, tibialis anterior, yeah. So it would be the antagonist, right? And then a bunch of the little muscles in our feet that keep all of our ankle bones in the relative position they're supposed to be in would all be stabilizers. To stabilize your ankle. Okay? So a second class lever is free. Not E-fur, but free. So a third class lever is a reverse where the fulcrum is still in the same place, but resistance and effort have changed positions. Right? And so that would be classically flexion of the forearm. So the fulcrum is the elbow joint created by the trochlear notch of the ulna and the trochlea of the humerus. Right? And then our prime mover would be this one, which is our biceps. The muscle underneath it, the brachialis, would be our synergist. The antagonist would be the triceps. And our stabilizers would be the muscles that stabilize the scapula, keep the scapula in place. And then what happens is, because the insertion of the biceps is on the radial tuberosity, you're picking up the weight of your arm, then the fulcrum is back here, the resistance is here, excuse me, the effort is here and the resistance is here. So, it's cool examples of lever systems. Okay, so you guys good with motor units at this point in time? So motor unit is all of the muscle cells innervated by a single motor nerve. And the importance is it's the level where the all or nothing principle occurs. A motor unit, the cells that make up a motor unit, the myofibrils that make up a cell, and the sarcomeres that make up the myofibril will either all contract fully or not contract at all. There is no such thing as a partial contraction. So the way a muscle, like the biceps, can do a partial contraction is the biceps is made up of many motor units. And if you only do a partial contraction, the brain only sends signals to part of the motor units. To do a full contraction, the brain has to send signals to all of the motor units. And that's what you worked on for the first part of your life. Learning how to walk, learning how to throw a ball, learning how to tie your shoes, learning how to write. Was your brain was working on creating the pattern of the amount of motor units you needed to contract to accomplish a specific task. So what PTs and OTs do in a clinical environment dealing with people that have had strokes is retraining people to go through that same stuff you went through as a small child to learn how to accomplish tasks. And different areas of the brain that have been undamaged are the ones that actually have to learn it and remember it. And so can you imagine having a stroke and not being able to remember how to tie your shoes? And having to relearn how to tie your shoes, which is what happens. Because the memory of that sequential event was what was damaged. All right. So if we go from a partial contraction to a full contraction, it's because the brain undergoes recruitment. And it sends signals to a greater number of motor units. So if we increase the number of motor units actively contracting, we convert a muscle from partial contraction to full contraction. And that process is called recruitment. Okay. That's what we talked about this week. So when we look at contractions, we can divide contractions into two types of contractions. So an isotonic contraction and an isometric contraction. So in isometric contractions, you have an increase in muscle tone, but the muscle doesn't physically shorten, largely because antagonistic muscles are pulling on one another. So we use isometric contractions to maintain body position. So how do I stand erect? My rectus abdominis has to contract on the front, and my erector spinae muscles are contracting on the back. And they're both pulling against the vertebral column and ribs. So that stabilizes my position. So if I want to convert from an isometric contraction to an isotonic contraction, then I would have to either contract my erector spinae harder, which would make me extend my vertebral column, or contract my rectus abdominis harder, which would make me flex my vertebral column. So we go through the day stabilizing body position by using isometric contractions a lot. Okay, so you can demonstrate it. So if you make a fist and then squeeze your fist, then you can feel the extensors have tension on them, and you can feel the flexors have tension on them. So once you do that, try to use your flexor carpi radialis and your flexor carpi ulnaris to go from this, which is an isometric contraction, to this, which is a isotonic contraction. Now you feel a bunch of tension on the back of your wrist, where you're actually pulling against tendons that have tension on them. So convert from isometric to isotonic contractions. Now when we look at isotonic contractions, we can divide them into two types of isotonic contractions: concentric isotonic contractions and eccentric isotonic contractions. So the first problem we have intuitively with this is the way we have traditionally defined the word contraction. So if I say contraction to you, you would all say the muscle is doing what? Shortening. Okay. So what we've done is we've defined the word contraction as a shortening of the muscle. Okay. So now, what if we took the word contraction and redefined it to what's actually going on within a muscle cell itself? So what's actually going on in a muscle cell that allows the muscle to shorten? Actin and myosin are combining, right? And we're using ATP to actually try to pull the actin toward the center of the sarcomere. So if we use the idea of a contraction as this interaction between actin and myosin, then we get out of that dilemma as contractions always mean shortening. Right? So in a concentric isotonic contraction, the overall length of a muscle decreases or shortens. In an eccentric isotonic contraction, the overall length of a muscle increases. But we're using the word contraction in both because it's referring to actin and myosin interacting. All right, so at any action that we just did, the prime mover and the synergist in flexion would be doing a concentric contraction. The antagonist, the triceps, is actually doing a eccentric contraction. So anytime we're looking at a joint, what we're saying is the triceps is just not doing nothing. It's actually got actin and myosin interacting. But because the biceps is creating more force, then even though actin and myosin are interacting in the triceps, the sarcomeres are actually being stretched by the force of the prime mover and the synergist. And you can do that. So do this. The triceps still has tension on it. It's just not flabby. It's actually got tension. Yeah. So when you do muscles and you're trying to build muscle mass, one of the things to do, like on a curl, is to bring it up with the weight. You can just let it fall like that, or you can drop it really slowly. So now that we're extending it with weight, the triceps is the prime mover, the biceps and the brachialis now are the antagonists. So now the triceps is doing a concentric contraction, the biceps and the brachialis are doing a eccentric contraction. And if you do that with real heavy weights, you tear up a bunch of sarcomeres because you're actually pulling them apart as they're trying to contract. And then 2 days later, your arm's going, oh my God, I can hardly move this arm, it hurts so bad. And then your body has to reconstruct all the sarcomeres and myofibrils. And what you do is you make more myofibrils and make the muscle bigger in diameter. And that's why you build muscle mass. You don't add new cells. You make existing cells bigger to tolerate the force that was put on. So if you want to build bulky muscle mass, either do it with anabolic steroids that make you make more myofibrils and make your muscles bigger, or you do it with force, or a combination. People do. Okay? All right. So don't forget your homework due tomorrow on your graphs. Now we're going to pick up the idea of tension